Welcome to the webinar, Skin in the Game, Get Safety Right, Non-Clinical Study Requirements for Dermal Therapies. Today we'd like to discuss an overview of dermal therapeutics, the safety studies needed to move a dermal product into clinical trials, and study considerations that need to be thought about when taking your dermal product from conception to first-in-human clinical trials. Overall, there's an estimated 1,200 companies worldwide that are trying to develop dermal therapies, and approximately 30 new companies are being launched annually with dermal products that are looking to be developed. These products include topical treatments, which could be creams, lotions, transdermal products, fillers, or therapeutic skincare. And commonly, the way that these are worked is to take existing drug products that have been administered by a different route of administration, such as injection, and reformulate them to allow dermal administration, which provides a method for patients to be more compliant, theoretically, with administration of the product. And so if that's the case, that it's a reformulated product, it would follow a 505(b)(2) or a more truncated pathway to IND approval. This is not to say, of course, that you would not be able to use a novel product that would only be administered dermally and follow the more traditional 505(b)(1) pathway for your IND. A number of dermal products contain small molecules, and while some do contain biologics, this discussion is going to be based mostly around the requirements for small molecules. The requirements for biologics are somewhat consistent with small molecules, with a few minor differences that would be best described with your individual groups that are helping put together your regulatory strategy for your dermal product. The metabolism of your small molecule is a central aspect to choosing your species selection for the small molecules. Generally, if you're doing products that are oral or intravenous, you would normally aim to use rats and dogs. However, in this case, because the skin is much more consistent between minipigs and humans, the non-clinical approach that's going to be described today is going to talk about using the minipig as your non-rodent species for describing how to get your product towards regulatory approval. Rodents, specifically generally rats, would be still needed to move your product to approval, as systemic toxicity is going to also need to be described, and we'll be talking about that as we move forward. So the non-clinical studies that would generally be required for dermal products would be single and repeated dose toxicity studies in the rats and the minipigs, with rats being systemic toxicity and the minipigs being local or topical treated toxicity, safety pharmacology, genotoxicity, skin irritation studies, skin sensitization, phototoxicity, and an eye irritation study, which will describe the bovine corneal opacity and permeability test, although some other alternatives are available. It's important to note, though, that if you are working with a 505(b)(2) pathway, some of these studies, such as genotoxicity, may have already been conducted to support the original indication of your drug under a different route of administration and may not need to be repeated. As discussed previously, the rodent studies are being used generally to understand the systemic toxicity of your drug. What happens if your drug gets into the circulation? Normally, most dermal products are based towards the skin, unless you're using transdermal patches, which would allow more systemic routes of administration. Therefore, for the rodent studies, we generally use subcutaneous administration to provide 100% bioavailability and mimic 100% skin penetration. The rodent studies are generally done in rats as described previously, but if the pharmacology is for mice, mice are an acceptable species to use for your rodent studies. The toxicology studies, as would be done for almost any small molecule, would include a non-GLP maximum tolerated dose and dose range finding study, and a GLP study of 14 or 28 days, depending on your initial clinical trial design. So the important thing to remember for the rat study, which will also be consistent for the Minipig studies, is that the animals need to be dosed to the maximum tolerated dose, the maximum feasible dose, or the limit dose. As most dermal products are being dosed at more than 1 gram to humans, the limit dose is 2,000 milligrams per kilogram per the ICH M3R2 guidance. The maximum feasible dose would be dependent on the solubility of the drug, dose volume limitations, or the maximum tolerated dose would be based on toxicity results. So all of these need to be considered when deciding how much total drug could be administered to your rodent studies. For the mini pigs, the first question is, which strain of mini pig do you want to use for your study? And what you'll notice from the picture here is regardless of the strains that are noted, the Gottingen, the Sinclair Nanopig, or the Hanford, they're all lighter in color. And the reason for this is that lighter colored skin makes it easier to help evaluate skin irritation potential in the animals. Skin irritation is going to be a combination of erythema or redness and edema or swelling. And so the lighter skin on these mini pigs will allow for an easier technical read on the erythema scores versus using a pig with a darker skin tone, such as a Yucatan. So the mini pig studies are going to be used to help understand the local toxicity of your drug. So you're administering your cream, lotion, patch to the skin itself, and you're looking to see how your drug is affecting the skin directly. The formulation that's going to be dosed should be consistent with what's going to be used in the clinical trials. And you need to dose over 10% body surface area, once again to the maximum tolerated dose, the maximum feasible dose, or the limit dose of 2,000 milligrams per kilogram. It's important to note that with mini pigs being quite large, the amount of test article that's going to be needed is going to be much bigger than what would normally be needed for any other types of small molecule development program. And so it's important to keep that in mind when you're starting to make your manufacturing for your drugs to support your mini pig safety studies. The dose that's going to be used, while it's going to be set up to that 2,000 milligrams per kilogram or 2 grams per kilo, that would be varied potentially by either the concentration of the drug, the percent of the active in the formulation, or by the volume with a single concentration of active in the formulation. So, for example, if your drug is intended to be used at a 0.5% in a cream or lotion, you would dose either 0.5, 1, 2% in the same volume across 10% body surface area, or you can dose a half, 1, and 2 grams per kilo of that 0.5% clinical formulation. to 10% body surface area. The important thing is no matter which you decide to do, you want to ensure that the entire 10% body surface area is receiving complete coverage of the dose. As with the rat studies, these toxicology studies would also involve a non-GLP maximum tolerated dose and dose range finding study, and then a GLP study of 14 or 28 days, depending on how you intend to dose in the clinic. But the important thing to remember for the Minipig dermal studies, unlike the rats, is that because it's being applied topically, one of the questions that the agency generally would have is there the possibility that your dermal product is going to be administered to broken skin? If it's absolutely never going to be applied to anything but intact skin, there's no need to manipulate the mini pigs prior to starting your dose administration. However, generally, it's assumed that patients are going to have nicks, cuts, scrapes on their skin, which could increase potential systemic penetration of your dermal product. Therefore, to look at the worst case scenario for your patients, you would generally do tape stripping or other types of abrading to remove the stratum corneum of the skin, the topmost layer, to maximize the systemic penetration of your dermal product. While this is not necessarily the way that it's going to be applied to your patients, this is providing the most conservative approach to safety so that the agency will understand the worst case scenario of what might happen to your patients if the drug is applied to skin that's not intact. To do tape stripping, generally you would apply the tape against the skin and pull it off to remove one layer at a time, and this would generally be done initially 20 to 30 times to remove the entire stratum corneum. But since the skin will start to regenerate over time, generally we would go back in once a week and do another 10 tape strips just to remove any stratum corneum that has started to be laid down as you go into a longer term study. This will allow the assurance that you're working on the broken skin or abraded skin for the length of the study. For safety pharmacology, which is consistent with any small molecule development that you would normally do, the three required safety pharmacology studies would be the central nervous system, the respiratory, and the cardiovascular system. Generally, the central nervous system studies are done in the rodent. Respiratory can be done in either the rodent or the non-rodent, and the cardiovascular studies would be done in the non-rodent species. Now, the respiratory and the cardiovascular studies can be combined into a single study, which is generally done in the non-rodent, but these can be standalone studies or they can be incorporated into your repeat dose toxicology studies. The important thing to remember, though, if doing the incorporation of the cardiovascular and respiratory into your non-rodent repeat dose toxicology studies, is that these are generally done with jacketed telemetry or some other type of non-implanted system, and that may impact the dermal application of your drug since the jackets would go on the outside of the mini pigs. So this needs to be thought about when putting together your safety pharmacology studies. Genotoxicity studies, the S2R1 guidance does allow for one in vitro, the AIMS test, and two in vivo studies to be performed as an alternative to the typical two in vitro and one in vivo study. Either way, these studies are allowing you to understand the potential for your drug to induce genotoxicity in humans. Skin irritation studies could be performed in either in vivo or in vitro methods. The in vitro assay that the FDA has accepted in the past is 3D reconstructed human epidermis model. The in vivo study that has been used traditionally is the skin irritation study in albino rabbits. Neither of these may be required to support your IND if you have enough dermal irritation information from dermal administration to your mini pigs. However, this is something that should be discussed with the FDA at a pre-IND meeting so that there's no question that something might be missing prior to your IND submission, because you would not like to have a reason to be put on a clinical hold before you have a chance for your IND to be accepted. Skin sensitization studies traditionally are done in albino guinea pigs using either the guinea pig maximization test or the Buehler method. In Europe and in the United States, for cosmetics, in vitro assays are used. And the FDA has noted that they will consider these, but their latest guidance from 2023 on the immunotoxic potential of pharmaceuticals notes that they currently accept the guinea pig maximization test. Although it's not as sensitive as the maximization assay, the Buehler method doesn't require adjuvants, which cause pain to the guinea pigs, and has been used consistently as an acceptable method for skin sensitization that has been accepted by the FDA. Another alternative method for skin sensitization would be the local lymph node assay, which is done in mice. This assay is specifically called out in the 2023 guidance as not recommended due to potential for false positives depending on the drug that's being used for the assay. However, if it's negative, the FDA will accept the LLNA results as a non-sensitization response. Phototoxicity is important because your patients theoretically are going to apply this drug to their skin and then eventually go out in the sunlight. And the question is whether or not your drug could potentially interact with sunlight to result in a toxic effect on the skin. So the first thing that's done per the S10 guidance for phototoxic potential is to look at the absorbance of the test article. And this is done in a chemical assay. So there's no in vitro or in vivo test involved to look at the molar extinction coefficient. If the molar extinction coefficient over the length of the absorbance of the light is less than 1,000, then there's no need to proceed with any other phototoxicity assays, and it's considered to be negative. If there's a positive result at any of these absorbance levels, an in vitro phototoxicity test with 3T3 fibroblasts is generally performed. If this is negative, then once again, you're finished, and you can note that there's no phototoxicity potential. If this test is positive, then there would be a requirement for in vivo phototoxicity testing, and the guidance provides some indication of ways that this can be performed, although it's not prescriptive and definitely should be discussed with the contract research organization that you're going to be working with to ensure that you can design an appropriate phototoxicity study to meet FDA requirements. The last test that needs to be performed is the bovine corneal opacity and permeability test. This is an ex vivo test, so it's using eyes from cows from slaughterhouses to determine the ocular toxicity. And this was used now to replace the in vivo rabbit ocular toxicity, or the DRAISE test. The in vivo rabbit ocular toxicity test was used previously by a number of cosmetics companies and resulted in a large number of issues from animal welfare groups and has therefore commonly been replaced. For this assay, The bovine corneas are dissected and treated with the dermal product directly for 10 minutes. And then the opacity and permeability are measured, and an in vitro irritancy score is determined, which allows identification of whether or not this product is going to potentially induce eye damage. And then this allows for the agency to understand the potential for the product to cause eye damage. The question a number of people might ask is, why is this important? And the answer is that you generally will apply this material onto your skin. The chance that you can touch your skin and then touch your eye is very high. And so this allows you to understand if there's any risk to the patient after touching their skin and touching their eye that they might transfer some materials to their eye and result in eye toxicity.
All right, thank you, Jeff, for that great presentation on the studies that are needed to help support a dermal therapeutic. At this point, we're going to change gears a little bit and talk about study considerations. When you place a safety study, and this applies to any safety study, not just dermal studies, you need to put it with a lab that has experience with that model. But with a dermal program, I think it is a little more important to be aware of the experience level of the particular lab, because Minipig studies, when you look at non-rodents overall, dogs and monkeys are by far used more frequently, with Minipigs being a distant third. So the amount of experience that a particular lab may have with Minipigs can be variable. But that means you need to pick a lab that has the experience in not only designing and executing, but also interpreting the data from a Minipig study. The Columbia site for Altasciences has run nearly 650 Minipig studies since 2015, with about a third of them involving dermal dosing. It has experience with the pigs that Jeff indicated that are involved in dermal, which are the Gottingen, Hanford, and Sinclair, as well as the Yucatan Minipig. So this site has extensive experience. And over the years, these studies have been submitted to the regulatory agencies, help supporting clinical trials, as well as supporting approval packages. Now, the FDA doesn't really dictate which breed you should use for your Minipig studies for dermal, which then means that the decision is up to the sponsors. So scientifically, all of these breeds provide a model of human skin that is more relevant than other non-rodent species. So then if the scientific aspects are met, then you can look at things other than scientific reasons for using one breed over another. One of those being size. Now the Sinclair Nanopig is the Sinclair Minipig. It's just been downsized and rebranded as of the end of 2022. So it was a 12-year effort to downsize the animal using selective breeding and nutritional management. With the breeding, it was basically smaller sows, smaller boars, smaller piglets, repeated, with some input from genotyping and looking at particular SNPs to help guide that breeding. In addition to that, there is nutritional management. This is not simply cutting the diet. This involved a lot of research into the caloric needs of the animals to make sure that they develop normally and that you wouldn't have any issues with abnormalities due to insufficient calories. So the combined effort of that selective breeding and the nutritional management was that the Sinclair Minipig was reduced in size anywhere from 30% at a young age to 60% at an older age from the normal Sinclair Minipig, making it smaller than the Gottingen and slightly larger than a beagle. Now, the Columbia site has generated historical control data for pathology data, as well as reference ranges for clinical pathology. Now, historical control is something that is needed when you have unusual findings. It is not always needed, but when you need it, you must have it. And it is now something that we have for the nanopig at the Columbia site. One of the important things to know with the nanopig is that there were no genetic alterations involved in this process. This is the Sinclair Minipig. just smaller and rebranded. So here we see an internal study where we took 8 males and 8 female nanopigs, starting at 3 months, and we followed them for 9 months, mimicking a chronic study that you would use for your approval of a novel therapeutic. As you can see early on, the nanopigs are slightly less than the Gottingen at 3 months, about 5 kilos for the nanopig and about 7 kilos for the Gottingen. They share a common body weight trajectory up through the next 4 or 5 months, but then you can see that there's a separation, that the nanopig growth rate plateaus more than the Gottingen. And at the end of the study, about 12 months of age, there's a substantial difference in body weight between the Gottingen and the nanopig. Another way to look at this is in this table, where we had the body weight at the start of a study, and I'm looking at a 9-month study here. So you have the 4 breeds there, vastly different starting body weights, from 5 kilo for the nanopig to 15 kilo for the Hanford. By the end of the study, though, there's a substantial difference. The nanopigs top out at about 15 kilograms, whereas your Hanfords will get up to about 60 kilograms. Now, as Jeff mentioned, you may have to dose up to the limit dose of 2 grams per kilogram. So doing the math, you can see that the amount of testicle needed for a nanopig is vastly less than that for the Yucatan and Hanford for sure, and almost half of that for what is needed for a Gottingen. So what that means to our sponsors is that if formulating the testicle or manufacturing the testicle is either expensive or timely, the nanopig represents a Minipig option that is accepted by the FDA, represents human skin as best you can for a non-clinical model, and it saves them on testicle. This webinar helped you to understand that dermal products have some unique aspects. There are some unique studies that are not noted for other routes of administration, such as the BCOP assay that Jeff outlined, but then also the fact that your pig study will be dosed by the clinical route, whereas your rodent study is being dosed by a route that is not the clinical route, which is unusual because usually the rule of thumb is you dose the animals by whatever route you're dosing in the clinic, not something that's applied in dermal space. Now, the studies that Jeff outlined have been used for multiple INDs over the years. And so it's very readily accepted by the regulatory agencies. But one thing I've learned over the years is that there is no one size fits all. One of the nice things that I've noted over the last 10, 15 years is that the regulatory bodies are amenable to discussion, scientific discussion, about finding alternative paths. So while the studies Jeff outlined are definitely something to write down and to bear in mind when developing your program for your particular molecule, if there are reasons why you feel one study or another may not be needed, take it to the FDA. Use your interact meeting, use your pre-IND meeting, or any of your interactions to discuss this with regulatory agencies as to why you feel that study may not be needed or may be done in a different way. The FDA is much more amenable now to finding alternative paths. Number one, reducing animals. Number two, reducing the cost and the timeline to get therapies to humans. When you're outsourcing your non-clinical studies, you need to make sure that that lab that you're placing your work with knows the ins and outs of running pig studies, because it's not the same as a dog study or a monkey study. Pig studies are unique. There are clinical signs in minipigs that you don't normally see with other animals. Most notably, at least to me, when I started running minipig studies, is the amount of eye discharge in pigs. It's a frequent thing that you'll see all the time, but if you're not used to it, it can be something that can be concerning. So you need to make sure that wherever you're placing the work, they are used to running the study, interpreting the data, to provide you with the best safety assessment that for your molecule. And then one thing that I think people may not realize is that the breed of mini pig that you are to use for your dermal product is not dictated. The FDA doesn't say use the Gottingen or the Hanford or the Sinclair. The FDA is agnostic to that. A case in point is, all those mini pig studies that have been run at the Columbia site have gone to the regulatory agency, and they've been in the different breeds. The FDA really doesn't care which breed it is, so it's up to you as a sponsor. You have options. You're not tied into one breed or another, so you can't really make a wrong choice when it comes to that. But you need to understand what are the implications of this breed over that breed. Primarily of that is going to be the size of the animal. And as I showed you, the nanopig represents an acceptable mini pig breed for dermal development that also saves you on test article usage.
All right, thank you all for attending the webinar. I think we have some questions in the chat. So I'm going to be the moderator. Jeff's going to be the respondent. So Jeff, the first question is, while 10% body surface area is common in dermal studies, I have seen 5 or 15% used. What determines whether or not you use, 10%, 5%, 15%?
It's a good question, and I've seen sponsors ask that before, but generally the agency is expecting 10% body surface area. And so if somebody is thinking about higher or lower amounts than 10%, it would definitely be recommended to have a pre-IND meeting with the FDA and get agency feedback before deciding, especially to go lower than 10% body surface area. If you choose to go higher, there's nothing from a regulatory perspective that's inherently wrong, but it may just mean that you're using more test article than you need to, and it's just going to, in the end, not really provide additional information that's going to give the agency that feeling of safety going into your clinical trials.
All right, so the next question is an interesting one. Is there any situation where you wouldn't use swine for a dermal study?
That is an interesting question because generally swine are considered to be the closest to human. And so I'm not usually seen where swine wouldn't be used as your non rodent species for a dermal study. I know that some individuals would like to run rodent studies as well, since rodent skin is thinner than human, and therefore you could potentially see worse findings or more exaggerated dermal findings than you would see in a mini pig. But I've not normally seen studies where pigs are not the non rodent species of interest.
And then we've got a few questions about study conduct. First one's a twofer. What are the potential sources of variability in dermal studies? And then how can consistency be maintained across technicians in, say, a 9-month study where you're dosing every day for 270 some days?
That's always a concern of a study director who's trying to manage a study from a dermal perspective. The most big variability is the technicians generally. And because, as you said, over a 9-month study, you're going to have different technicians providing input to DRE scoring, which is usually the most variable. And so the best way to reduce that variability is to have a good training program that allows all of the technicians to work together to figure out exactly how the scoring is going to be maintained and to not hesitate to question each other back and forth over time to verify that the scores are going to be consistent. In addition, it's always good to ensure that the technicians are looking at the scores over time for their studies so that if they're calling a 1 and the day before was a 4 from an erythema perspective or an edema perspective, the chance that things are going to move that much over a 24-hour period is generally low. And so checking to make sure that scores, not to check that the scores are um, appropriate on a daily basis per se, but to make sure that there's not a huge amount of variability on a day-to-day basis with the material jumping scores too drastically every day to ensure that consistency between the technicians' calls. The other sources of variability, it could be related to the test article. Hopefully, the test article is some sort of a homogeneous material that's coming, whether it's a cream or a lotion or some sort of a liquid that's going to be administered. You want to ensure that it's going to spread evenly and, thin, well, as thin as possible. If you have to go to 2 grams per kilo, things can get a little thick, but you want to try and make sure that you have an even spread of the material across the 10% body surface area so that you're not getting areas of the body that are not getting a full spread of the test article, because that could also have an effect on the amount of material per centimeter squared that's getting exposed to the test article.
Hey, so you mentioned scoring. So a question here is, how frequently should the dermal observations be recorded? Multiple times in a day, twice a day, once a week? And then along with that, at what point is there a rule of thumb? At what point does something become an adverse reaction?
Okay, so typically for dermal scoring, once a day should be adequate. The material is going to generally sit on the skin for 24 hours. So doing dermal scoring once a day for the first week, and then if the material is not causing too many concerns, then scoring once a week for the remainder of the study, depending on the length of the study, should be adequate. Now, if dermal scores start to go awry early on in the study, you may need to amend the protocol to get more frequent dermal observations daily for 2 weeks or 3 weeks until you can start to see that either things calm down or you need to make other modifications to the study. So, when it comes to the question of adversity, that is sort of a question that's going to be addressed by the study director in conjunction with the veterinary staff and the technicians, because unfortunately, it's not a cut and dry answer. Um, it's easy if you see a score of 4, the most, you know, horrible things that could happen theoretically from an edema or erythema perspective. It's easy to say if you see a score of 4, you have to do something. The problem comes in when you start to see scores of 2 and 3 as the erythema and edema start getting worse. It's not necessarily instantaneously adverse. And it's a question of experience of the study director and a discussion with the veterinarians to see whether or not the animals are in pain or experiencing some sort of discomfort. Just having redness or just having the skin show edema, have swelling of the skin, isn't necessarily by itself adverse. It's when the animals start to show that discomfort or pain that you need to then have some sort of veterinary intervention and or dose holiday so that the animals are allowed to recover and not continue in a state of pain.
Once again, thank you all for joining us for this webinar. If we were not able to get to your question, don't worry, we will be responding to you directly so that you will get the answers that you need. And again, thank you for your time.